HIV Life Cycle by Erin, Boland. Attachment. The life cycle of the HIV virus begins with attachment of the HIV virus to the T lymphocyte host cell. With attachment, GP120 proteins from the HIV virus bind to a CD4 receptor site on the surface of the host T lymphocyte cell, which then draws in closer. The GP120 molecule then binds to one of two co-receptors, either CXCR4 or CCR5. This releases the GP41 molecule, which pierces the membrane of the host cell, allowing the HIV virus to penetrate and release its capsid into the cell. Entry and encoding. Then the next step is encoding, wherein the genome from the nucleocapsid is removed. Cellular enzymes strip away the protein capsid, releasing the RNA integrase protease and reverse transcriptase. Reverse transcription. The next step is reverse transcription. On the inside, the reverse transcriptase takes the HIV RNA, uses it to make a single strand of DNA, then decreates the RNA and uses that single strand of DNA to make a second strand of DNA, forming a DNA helix with the genetic information of HIV. Reverse transcriptase has thus converted the single-stranded HIV RNA into double-stranded HIV DNA. Integration of provirus. The DNA then migrates to the nucleus of the host T lymphocyte cell. In this stage, the viral enzyme integrates, splits the DNA of the host cell, and incorporates the viral DNA molecule into the cell's DNA at a random site. This completes the provirus, the integrated HIV DNA that's now part of the host cell's nuclear material. This provirus is now inherited as a gene for all the descendants of the host cell. Production of mature virions that are infectious. When the host cell receives a signal to become active, the provirus uses the host enzyme RNA polymerase to create copies of the HIV genomic material, as well as shorter strands of messenger RNA. The virus DNA codes for virus RNA in different proteins. The HIV enzyme protease cuts long chains of HIV proteins into smaller individual proteins. As the smaller HIV proteins come together with copies of HIV's RNA genetic material, a new virus particle is assembled, and the capsid for the HIV virus and enzymes is formed. In the final stage, a new HIV virus pinches off from the host cell, taking part of the cell's membrane with it as its envelope. This new virus now moves through the extracellular environment to begin the life cycle anew.